Today's experiment is estimation of cholesterol in blood sample, or title may be as enzymatic calorimetric method, quantitative determination of cholesterol in serum or plasma. This is the title of today's experiment, and we need to estimate the total cholesterol. Here it's mean cholesterol estimation means we are determining total cholesterol in the blood, and this experiment is included in the blood profile. Here we will estimate total cholesterol. This is quantitative determination of cholesterol in serum or plasma, and the methodology is enzymatic colorimetric method. Enzymatic means we will use enzymes for estimation of cholesterol in the serum, or colorimetric method means there will be formation of colored compounds in the end of this biochemical reaction, and that colored compound will be estimated by using spectrophotometer. So we need spectrophotometer for the estimation of cholesterol. Spectrophotometer for the estimation of cholesterol in the serum or blood plasma. Now mostly when we proceed the estimation of cholesterol, estimation of triglycerides, estimation of low density lipoproteins, estimation of high density lipoproteins, estimation of very low density lipoproteins, estimation of ALT, estimation of AST, estimation of phosphatases enzymes, or when we estimate the heart enzymes, when we estimate the kidney function test. In all these biochemical tests, mostly the tests of blood profile are proceeded with the help of kits. Kits are the commercially prepared reagents which are prepared by different diagnostic uh, companies which prepare different reagents for different biochemical tests. We use those kits. So here in today's experiment, we will use a kit that is prepared by MTD Diagnostics. This is a company which is preparing the reagents and providing them to the diagnostic labs. So in today's experiment, we will use the kit of MTD Diagnostic Company, and in this kit we have all those reagents and enzymes which are required to proceed this experiment, to proceed this biochemical test. So we should have a kit which is having all the reagents and enzymes, and other thing is we should have spectrophotometer. We should have spectrophotometer. So we need this kit and this apparatus for the estimation of cholesterol in blood sample or blood plasma. So first of all, we discuss what is the principle of this experiment, how we can estimate the cholesterol in the blood serum. The principle is it would represent which biochemical actions will be proceeded in the test tube when we proceed the estimation of cholesterol in the blood plasma. First of all, uh, determination of cholesterol after enzymatic hydrolysis and oxidation. The calorimetric indicator is quinone amine. Calorimetric indicator means this is the last colored compound that is called quinone amine, and This colored compound will be utilized for the estimation of cholesterol in our blood sample. So the colored product, which is formed after all these biochemical actions, that will be quinone amine. This is here. This is the final product which will be formed after all biochemical actions in our blood serum. And then we will estimate this quinone amine. And on the basis of the quantity of quinone amine, we can estimate what is the concentration of cholesterol in the blood plasma. So the colorimetric indicator is quinone amine. It will be colored product after all these biochemical actions, which is generated from 4 amino antipyrine and phenol by hydrogen peroxide under the catalytic action of peroxidase enzyme. This is the enzyme which we need to proceed this experiment. Now, the first biochemical reaction which will be proceeded in the test tube after adding the reagents in the blood serum or in the blood plasma. So, first of all, in the serum or in the blood plasma, cholesterol will be present in the form of ester, that is called cholesterol ester. And this ester reacts with water molecule in the presence of enzyme CHE. Anyone can abbreviate the CHE enzyme?
Sir, cholesterol esterase.
CHE.
Carbohydroxyesterase.
Cholesterol esterase. The enzyme is that is written here cholesterol esterase. The enzyme will be CHE. So we have cholesterol ester here, and this cholesterol ester will be broken down into two products. One is cholesterol and other is fatty acid. So cholesterol is present in the form of ester, and that ester is formed with the fatty acid. It will be present in the blood serum, and enzyme is here cholesterol esterase. This enzyme will be responsible to proceed this biochemical reaction. Right now, we have cholesterol and fatty acids in the test tube. Now, we need to estimate this cholesterol. So, this cholesterol in next biochemical reaction, it would react with oxygen molecule in the presence of another enzyme. We have CHO, and CHO, this stands for the enzyme that is here, CHO cholesterol oxidase. So this enzyme will be present in the kit. We will use the reagent in the test tube, and all these chemicals will be provided by this MTD diagnostic company, or there are different companies which are providing us the kits for different biochemical actions. Human kit है, ठीक है, तो different kits होंगी. Human की होती है, और MTD की है, यूनिवर्सल की है, और क्रिसेंट की है. तो इस तरह से देर विल बी डिफरेंट कंपनीज विच आर प्रोवाइडिंग द किट्स फॉर diagnostic tests. So here we are having cholesterol oxidase enzyme, and this cholesterol oxidase enzyme would react with the this oxygen will react with the cholesterol, and it would make cholesterol 3,1. This will be the product, and now we have cholesterol 3,1, and the required product is hydrogen peroxide. There will be two products. One is cholesterol 3,1, and other product is hydrogen peroxide. Now, if we have high concentration of cholesterol in the blood, it's mean there will be formation of high concentration of hydrogen peroxide. And if we have low concentration of cholesterol in the blood serum, there will be formation of low quantity of hydrogen peroxide. And in the next biochemical reaction, this hydrogen peroxide will be used, and this hydrogen peroxide reacts with 4 amino antipyrine. 4 amino antipyrine is not present in the serum. It will be added in the biochemical reaction, and it will be present in the kit. It will be present with the enzymes that is here. it will be provided from the kit, and we add this 4 amino antipyrine in the reaction mixture. Now hydrogen peroxide is reacting with 4 amino antipyrine, and again phenol is not present in the serum. It will be provided from uh, the reagent one, which we are adding in the uh, test tube. So here we are having the cholesterol as well, phenol as well. This phenol will be provided here. And now hydrogen peroxide reacting with 4 amino antipyrine and phenol group, and enzyme will be POD. Enzyme is here, that is POD. POD stand for peroxidase, that is here. We are having peroxidase enzyme in the kit as well. And in the presence of this enzyme, hydrogen peroxide reacting with 4 amino antipyrine and phenol, product will be quinone amine. This will be the final product, quinone amine. Now it will be colored, and we will place this reagent in the spectrophotometer. And when we place this reagent in spectrophotometer, it would absorb the light of specific wavelength, and then we read the absorbance, how much light is absorbed by this quinone amine in the spectrophotometer. As its concentration will be high, it would absorb more light. If its concentration is less, it would absorb less quantity of light, and there will be high or low absorbance. On the basis of absorbance, we can estimate what will be the concentration of cholesterol in the blood plasma. Now we are having quinone amine, and we will place this in the spectrophotometer, and you will estimate the quantity of cholesterol. Now the specimen, if we need to store the blood sample for 7 days, you will place it at 20 to 25 degrees Celsius, uh, or you can place it for at 4 to 8 degrees Celsius. But if you need to place your sample solution for long time, or need to store your sample for a long time, it will be blood sample or serum or plasma, right? You can store it for 3 months, but the temperature will be low. You need to place the sample at minus 20 degrees Celsius if you need to place the sample or store the sample for 3 months. Now the reagents composition, what will be the composition which will be provided by the uh, kit diagnostic or different diagnostic companies. Here MTD diagnostic kit would have two reagents. One reagent will be called as R1. It would have all the buffers, it would have all the enzymes, it would have all the reagents and chemicals which are required to estimate the cholesterol in the blood serum. And there will be another reagent that is called R2, that is it will be standard solution of cholesterol, and every solution will be standard if its concentration is known. And R2 will be the standard solution of cholesterol, and its concentration will be 20 milligram per deciliter. Now in R1, there will be goods buffer. This is the composition of R1. We need to use all these reagents, and its concentration will be 50 millimole per liter. And the second is, that is phenol, which is required in the biochemical reaction to make the quinone amine, that is 5 millimole per liter. This is the concentration. And next one is 4 amino antipyrine. Its concentration is 0.3 millimole per liter. Cholesterol acidates, it will be 200 units per liter. Cholesterol oxidase, it will be 100 units per liter. Peroxidase, it will be 3K, means 3,000 units per liter. This is the concentration of enzymes which will be present in the reagent one. All these reagents and chemicals and enzymes will be available in one bottle, which will be labeled as R1. Different companies would provide us the manuals and procedures with their kits. So here we are using the MTD kits, and we are using the manual of MTD diagnostics. If you are using the kit of some other companies, you will follow the instructions which are provided on the manual page of that company. So right now we move with the procedure how we can proceed this experiment, what will be the procedure, what will be the steps. Now here the reagent preparation, what will be the procedure how you can estimate the cholesterol in the blood. So first of all, sample start, the reagents are ready to use. Store the kit at 2 to 8 degrees Celsius. When you are not using it, you need to place it in refrigerator. The reagent is stable until the expiration date is as indicated when the stored at the recommended conditions. Discard if appear signs of presence of उसके बाद यहाँ से हमारा प्रोसीजर जब स्टार्ट हो रहा है, required materials which are not provided with the kit, that is general laboratory equipments like gloves, lab coat, and instruments like spectrophotometer. Now, what will be the procedure? How we can proceed this experiment? So, first of all, you will turn on the spectrophotometer and you will set the wavelength of spectrophotometer at 546 nanometer. It will be the lambda max of quinone amine. It will be lambda max of quinone amine, which is formed here, which is formed here in the test tube, the colored product. Lambda max means this wavelength will be the specific wavelength at which this quinone amine would provide us the maximum absorbance. Other wavelengths are absorbed by this quinolamine, but those wavelengths are not maximum absorbed. But 546 is maximum absorbed by this quinolamine. That's why 546 nanometer is called the lambda max of quinolamine. Now, the instrument with the spectrophotometer, we need cuvette and it should be 1 centimeter. Temperature that should be 37 degrees Celsius, and you will proceed it with the blank reagent. Now, how will you proceed the experiment? How will you prepare the test tubes? You need to take three test tubes. You will take three test tubes. One test tube will be labeled as blank. In blank test tube, you will not add any serum or plasma or standard. In blank test tube, you will add only distilled water. And the other test tube, second test tube, that will be labeled as standard. In standard test tube, you will use standard of cholesterol, which was labeled as R2. And it is having cholesterol and its concentration is 200 milligram per deciliter. And the third test tube you need to proceed this experiment that will be labeled as sample. Now we are having reagents R1, R2, and serum, and the last one is distilled water. Here we are writing distilled water, then we are having serum, and then we are having R1, and we are having R2. These are the reagents we require, and the third R2 is the standard. Now, in blank test tube, you will add 10 microliter distilled water. 10 microliter. You need to add 10 microliter distilled water in blank test tube. It will be 10. And No serum will be added in blank test tube, and you will add 1000 microliter, the R1 in blank test tube. That will be 1000 microliter R1, 1000 microliter. You will add R1 in blank test tube, and no R2. We don't need to add the standard. R2 is the standard solution. In blank test tube, you will add 10 microliter distilled water. Now in standard test tube, you will add no distilled water. In standard test tube, no serum. In standard test tube, you will add 1000 microliter R1 reagent. 1000 microliter R1 reagent here. And in standard test tube, you will add 10 microliter standard, that is R1. Now in the third test tube, sample test tube, you will add no distilled water in sample test tube. Third test tube is the sample test tube. You will add no distilled water, but in sample test tube you will add 10 microliter serum. This is serum, and you will add in sample test tube 1000 microliter R1, and in sample test tube no R2 will be added because R2 is the standard. Now you will place these three test tubes for 5 minutes at 37 degrees Celsius, and then after 5 minutes you will place these three test tubes or these three mixtures in the spectrophotometer for absorbance. First of all, you will place the blank in spectrophotometer and you will read the absorbance. Whatever the absorbance of the blank, you will make it zero. And in second step, you will take the absorbance of standard, and whatever the absorbance of standard you will write it, and then you will place the sample in the spectrophotometer and you will read the absorbance at 546 nanometer. Now you have three absorbances: absorbance of blank, absorbance of standard, absorbance of sample. Now you need to turn off the spectrophotometer and you will use the formula for estimation of cholesterol in your given sample solution. Now calculation with the standard. How will you calculate the concentration of cholesterol? Here we have formula. This is concentration of sample. Concentration of what? Concentration of cholesterol. Sample cholesterol concentration. This is cholesterol concentration. Now you are having the formula absorbance of sample, absorbance of standard, A for absorbance, into concentration of standard. Concentration of standard is here. Now you will put the number, you will put the value of absorbance. Let's say, suppose we have the absorbance of sample at 546 nanometer is uh, 0.05, and we are having the absorbance of standard, let's say, that is 0.05. 0.03. Now you are having 0.03 absorbance of standard, but here we need to multiply it with concentration of standard. Multiply it with concentration of standard. Multiply with concentration of standard. The concentration of standard is 200 milligram per deciliter. Now multiply it, and let's say answer is, we are having the answer. Answer is 333.33. Answer is 333.33. This is the answer. It's mean we are having 333.33 milligram per deciliter cholesterol in the blood serum of patient. 333.3 milligram per deciliter. Now what should be 333.3 milligram per deciliter? Now what should be the expected value of cholesterol in the blood serum, that is desirable? This is the ideal, that is 200 milligram per deciliter. It will be ideal if the patient having the blood cholesterol level 200 milligram per deciliter, it's good. But if the patient having the cholesterol level in the blood more than 200 degrees Celsius, in between 200 to 240 milligram per deciliter, it's mean he is on borderline risk, right? And if the patient having more than 240 milligram per deciliter, it's mean the patient is on high risk. Right now we are having the answer, that is 333.3 milligram per deciliter. It is more than the required concentration. It is more than 200 milligram. It's mean the patient is on high risk because it is more than 240 milligram per deciliter. So doctor would recommend him the medication that is that would decrease the cholesterol level in blood serum, and he would recommend him exercise, and he would provide him a specific diet plan to control the cholesterol level. This is how we can estimate the blood cholesterol level in the serum. Any question up to this?